Hi everyone, this is Scott Reeves here again for another FSN 328 lecture. And so this is kind of part two of the one we did before. So this is part two of carbohydrate lecture 2, I guess. And we went through some of the first part of this outline last time. We talked about carbohydrate digestion, the enzymes in their substrates. And now we're going to move into absorption of monosaccharides. And that'll start here. So one of the first questions is, how do hexoses enter cells? And the hexoses that we're going to talk mostly about would be glucose, fructose, and galactose. And remember, they're water-soluble molecules. So in order to go through a lipid bilayer, they need assistance from proteins. And we're going to call those proteins transporters. And there's two families of these transporters that we're going to talk about, the GLUT family and the SGLT family. So let's go ahead and start with that. Get my notes so I don't forget too much. So the GLUT family, and that GLUT stands for glucose transporters. And there are approximately 12 of these proteins that are considered GLUTs in this family. And they are integral membrane proteins, so they span that lipid bilayer, and that allows them to move glucose. They use the transport mechanism of facilitated diffusion. Right, so they transport D-glucose down its concentration gradient. So if there's a lot of glucose outside the cell, the glucose will run through a GLUT into the cell. If there's a lot of glucose inside the cell, it can run out of the cell. So the glucose can efflux depending on the gradient or amounts of glucose on either side of that cell membrane. So we'll talk about some of those characteristics as well as we go through this. So this slide shows a model for the structural orientation of the GLUTs. So you've probably seen this in another class, I'm guessing in biochemistry, where you have this diagram that shows the structure of a protein that's embedded in the lipid bilayer. So this would be the plasma membrane containing a GLUT. Now all of the GLUTs look fairly similar, so this diagram is meant to represent all of the GLUTs. And if we look at some of the things that are shown here, here's the amine group of the first amino acid, so this would be the amino terminus of the GLUT protein. And you can see it kind of heads towards the lipid bilayer. So sorry, we're intracellular here. This is inside the cell. Here's outside the cell. So the protein sequence starts here, first amino acid here, and then it goes through the lipid bilayer using an alpha helix. And remember that alpha helix structure is stabilized by bonds between parts of these amino acids to form that stable helix structure. And if I remember right, there's about 12 or 15 amino acids in these alpha helices in order to get through a typical lipid bilayer. So it kind of meanders through there, and then we have an extracellular loop. And then it comes back into the membrane. Remember, this is the same protein. Goes through the membrane, and each time it goes through the membrane, these structures are called transmembrane segments, or TMs. So these transmembrane segments contain mostly hydrophobic amino acids that can form these helix structures. So it goes through, then we have another intracellular loop, another transmembrane, outside loop, extracellular loop, back and forth, essentially forming 12 of these transmembrane segments. And that's pretty typical for a transport protein. Okay, so there's 12 of these transmembrane segments, but you notice there's loops on the inside and the outside of this protein. Now, some of these amino acids on the outside would have carbohydrates attached. This doesn't show it, but there might be a carbohydrate chain attached here, maybe some of these loops out here. Remember, that would be on the outside of this protein. The inside of the protein wouldn't be glycosylated. We talked about that when we talked about lipid bilayers and characteristics of proteins that are inside lipid bilayers. Right? So how did this protein get here? Well, it went through the secretory pathway, and then it was embedded in this membrane so that it can transport glucose. In this case, glucose in either direction because it's facilitated diffusion. Let's see. Some helices form hydrophobic pockets here. So there's different characteristics of these proteins, but the bottom line is down at the other end, here is the carboxylic acid group or the carboxyl terminus of this protein. So this is one GLUT protein in this membrane. Now, some of these GLUT transporters might work together. In other words, there might be a group of these, or either 2 or 4 of these together to allow glucose to go in and out. But they're approximately 500 to 500, about 500 amino acids long, each of the GLUTs. Okay, so that's basic overall structure. And all cells express at least one GLUT form. Okay? So this table shows the 12 GLUT family members, starting with 1 through 12, and their number is basically typically based on when they were discovered. So this protein, GLUT1, was discovered first all the way to 12. I remember a few years ago, they discovered a few more of these. So this list has grown. So there's a GLUT protein on every cell because every cell can use glucose for energy. So even though their sequences are similar enough to put them in the same family, they do have some distinct differences. And one of them is tissue specificity. So if you notice, for example, GLUT1 is expressed on red blood cells, the blood-brain barrier, the placenta, and fetal tissues in general. So GLUT1 is relatively widespread. Look at GLUT2. It's in liver cells, beta cells of the pancreas, kidney, small intestines. So you can see that these GLUTs are distributed throughout the body, but you tend to have one type of GLUT in certain tissues, and other tissues might have a different type of GLUT. Okay, so you can see there's a long list here. So I want you to put stars by GLUT2, GLUT4, GLUT5. GLUT2, GLUT4, GLUT5. Those are the ones that you need to know more details about. For example, you need to know that GLUT2 is not regulated by insulin, but you need to know that it's in liver cells, beta cells, and the small intestinal cells. GLUT4 is different. It's got an asterisk. It's written in red because GLUT4 is the only GLUT that is regulated by insulin. We've talked about GLUT4 before and how it works. But for us, we're going to say GLUT4 is in muscle cells and fat cells. Muscle cells and fat cells. When insulin goes up, their GLUT4 activity increases and they can facilitate the transport of more glucose into the cell. And here's GLUT5. GLUT5 is not regulated by insulin. It's in intestinal cells, and that's the only one you need to remember for GLUT5. Okay, and we're not going to talk about the other ones on this list. Sorry, I yawned. Okay, so those are the GLUTs. The next family is the family of SGLT. We're going to talk specifically about SGLT1. Okay, so these are sodium and glucose cotransporters. So they move both sodium and glucose across membranes. There's no sequence homology to the GLUT proteins. So they also transport glucose, but they are not similar in sequence to the GLUTs. The SGLT, they're integral membrane proteins. They span membranes, but they also have a sodium binding site. So they have both binding sites. So this is what we looked at before, and this diagram was actually done a long time ago in 1991. And they set up this kind of map of the structure of SGLT1, and you can see right away it has a lot of similarities to the GLUTs in its overall structure orientation. 12 transmembrane segments, same thing. So these would be alpha helices. Here's the amino terminus. This protein goes back and forth, back and forth. CHO is a carbohydrate site, so this would be a glycosylation site, one of these extracellular loops. And over here, here's the carboxylic acid group of the last amino acid. Now, SGLT1s tend to be bigger than GLUTs. SGLTs tend to be about 650 amino acids long. An easy way to remember that would be, well, maybe it's because they also have a sodium binding site in addition to being able to recognize and transport glucose, so maybe they need to be a little bit bigger because of that. Okay, so they have some things in common, right? They can both recognize glucose. They can transport glucose. But one thing to keep in mind is their transport mechanism is different. SGLTs work by indirect active transport, so they tend to be unidirectional, and that's different from the GLUTs that use facilitated diffusion. Okay, there's at least two of these SGLTs, and we're going to talk about SGLT1. So anytime you write SGLT, you want to remember that that stands for sodium glucose transporters. So you're going to write SGLT1. Okay, what else? Let's put these two together now. Let's look at the GLUTs and SGLT1. And we're going to look at it in the context of hexose transport into enterocytes. So we're going to spend a few minutes on this slide. This is a single enterocyte, brush border membrane, basolateral membrane, portal vein. Okay. If we look over here, we can see the monosaccharides, glucose, galactose, fructose, and we can see that SGLT1 is in the brush border membrane of enterocytes. GLUT2 is in the basolateral membrane of enterocytes, and GLUT5 is in the brush border membrane of enterocytes. But you can see that the monosaccharides use two different transporters to go into the cell and one transporter to go out. Okay, so I'll give you a basic overview of what's happening here, and then let's go through the steps of this process. So for example, here we see two sodium ions binding to a site on SGLT1, and then glucose or galactose binding. One of those binds, and then the monosaccharide and the sodium goes into the cell. The monosaccharides will run down their concentration gradient through GLUT2, through facilitated diffusion. The sodium, once it goes in, is quickly exported through the sodium-potassium pump, aka the sodium-potassium ATPase. It's called an ATPase because it hydrolyzes ATP in order to use the energy from that process to quickly and efficiently export sodium and import potassium. Most important part of that is it is exporting sodium. And so if we look at the way this is set up, the sodium and glucose or galactose absorption is coupled. It's coupled to sodium, and sodium uses energy in this process. So in an indirect manner, glucose and galactose absorption is indirect active transport. Fructose, on the other hand, goes through GLUT5 through facilitated diffusion, and then goes down. It can go down its concentration gradient through GLUT2. So let's go through the steps. Let's do glucose absorption first. So in step 1, sodium binds SGLT1. Sodium binds and changes the affinity of SGLT1. Sorry, let me try that again. Sodium binds SGLT1, changes its conformation. That increases the affinity of SGLT1 for glucose. Glucose binds. Both sodium and glucose are transported into the cell. Sodium is quickly exported through the sodium-potassium pump. Therefore, the sodium gradient is maintained. Remember, sodium is the major extracellular ion, so most sodium in the body is extracellular. And our body uses a lot of energy in order to maintain that gradient. The sodium gradient is important for things like glucose absorption, but also things like muscular contraction and neuron stimulation. Remember, that sodium gradient is involved in a lot of processes. I think it's estimated that every day we use about 1/3 of our ATP to maintain the sodium gradient. Okay, so it's the sodium gradient that is driving glucose absorption. That's an important point. Once glucose goes into the cell, the glucose will go through GLUT2 into the portal vein by facilitated diffusion, run down its concentration gradient. Okay, so sodium quickly goes in, it starts to accumulate, then it goes out through GLUT2. Okay? And in your notes, you can just say galactose is absorbed by the same mechanism as glucose. So sodium binds, change in affinity, galactose goes in, sodium's exported, galactose goes through GLUT2 into the portal vein and onto the liver. So that's hopefully pretty simple. So SGLT1 is specific for those monosaccharides. Remember, it'll transport either glucose or galactose, not at the same time. And it's on the brush border membrane, and it's unidirectional, indirect active transport. Now, fructose is different. Fructose uses GLUT5. GLUT5, even though it says glucose transporter, is actually a fructose transporter. Doesn't transport glucose, at least not to any significant amount. So GLUT5 is considered a fructose transporter, but it does use facilitated diffusion. So fructose binds, fructose goes through GLUT5 into the cell. Now, as fructose accumulates, the fructose can either go through GLUT2 or the fructose can go back out GLUT5 because it is bidirectional, facilitated diffusion. Because of that, fructose absorption is slower than glucose or galactose, because some of it actually effluxes back into the lumen of the small intestine. Sometimes that fructose is reabsorbed and ends up in the enterocyte and then into the blood. But if this system is overwhelmed, that fructose can pass through the small intestine. Remember, this is in the small intestine. If a person consumes a significant amount of fructose at the same time, sometimes it's not absorbed fast enough in the small intestine, and some of that fructose goes into the large intestine. In the large intestine, the fructose is metabolized by bacteria, and it can cause osmotic conditions that result in diarrhea. Sometimes a person feels bloated or just uncomfortable, and sometimes they'll think it's something else that's causing it. Some people might even think that it's gluten that is the problem, but sometimes too much fructose. And kind of a ballpark number there is about 50 grams of fructose is considered to be an amount that commonly causes some GI discomfort in people. Remember, that fructose can come from, it could be, for example, from high fructose corn syrup, it could come from sucrose, it could come from a variety of different foods, but I'd say in general it's probably either sucrose or high fructose corn syrup that can cause that. So that's why fructose is a little bit different. But then on the basolateral membrane, they all go through GLUT2 into the portal vein. Okay, so you'd want to practice drawing this diagram and explaining the process of absorption for each of the monosaccharides, including their mechanisms of absorption. Okay, so now let's kind of follow this through. Remember, we're on enterocytes here, but now these monosaccharides have gone into the blood. And in this case, the monosaccharides are now in the portal vein, and the monosaccharides would go into that circulatory system that's taking them towards the liver. Okay, so in this case, let's look at what happens to each of the monosaccharides. And I'll try to give this to you in a way that you can take notes on this. Let's talk about glucose first. So this would be like postprandial tissue uptake of the monosaccharide. So postprandial tissue uptake of the monosaccharides. So let's do glucose first. After a meal, approximately 34% of the glucose is absorbed by the liver, approximately 34%. The rest of it goes into the circulation. So after a meal, about 33% of glucose is absorbed by red blood cells and the central nervous system. About 33% is absorbed by red blood cells in the central nervous system. About 33% of the glucose is absorbed by muscle and fat cells. So you can see we have kind of three categories of tissues where that glucose ends up. About a third of it ends up in the liver. We're going to say 34%, about 33% in red blood cells and central nervous system, and about 33% in the muscle and fat cells. And you can think of it as the reason the liver doesn't take up all of the glucose is because other tissues rely on glucose. So the liver lets most of it go by. Now let's look at fructose. That fructose goes into the portal vein, to the liver, and approximately 100% of fructose is absorbed and metabolized by the liver. Galactose is the same as fructose. Approximately 100% of the galactose from a meal enters the liver and is metabolized by the liver. So that's another indicator that glucose is important to all the tissues, whereas fructose and galactose is not. Okay? So now let's start talking about how blood glucose is regulated. So this is the nutritional or physiologic significance of, we're going to be talking about the monosaccharides. So this is regulation of blood glucose levels. Let's talk about how it's regulated metabolically and then also for hormonal regulation. So metabolic regulation, blood glucose is tightly regulated. It's one of the most tightly regulated concentrations of a solution in the body. And I don't think I have that here. The fasting blood glucose concentration is typically 70 to 110 milligrams per deciliter. So the fasting blood glucose concentration is between 70 to 110 milligrams per deciliter. If it goes too low, our body responds. If it goes too high, our body responds. So it's important that that blood glucose is regulated even in between meals. And one of the reasons is because the brain depends exclusively on glucose under normal conditions. And we've talked about that a few times. Now, there is not a significant amount of glucose in the blood consistently. So if you took that concentration of glucose in a fasting scenario and you multiplied that out by the amount of the amount of blood that our body has, that would only give you about 16 grams of glucose in the blood glucose pool at any given time. Remember, this is fasting, so most times during the day. About 16 grams of glucose is traveling through the blood. That's not much. And so what that means is that most of the glucose is actually in cells, including the liver, so we'll talk about that. Okay? The other thing that we need to think about and remember is that the liver is the only tissue that will absorb glucose, but then put glucose back into the blood when the blood glucose starts to drop. So, you know, in normal conditions, the liver is basically putting out more glucose than it's absorbing. And one of the important relationships there is the liver can provide glucose to other tissues, but especially the brain and central nervous system. And overall in a day, the adult human brain tends to use about 140 grams of glucose a day, something like that, 125 to 140 grams of glucose a day. So that's one of the reasons the RDA is set at 130 grams a day. It's approximately the amount of glucose that the brain uses. So any other activity, like physical activity, that requires glucose, we need to consume over and above that, obviously. Okay? Let's talk about how it's regulated. So this is metabolic regulation, for example, the liver and tissue uptake and phosphorylation of glucose. So the first part of this that we're going to talk about is shown here. Here we have, for example, cell membrane. Remember, this will be glucose going into a cell from the blood. We're going to talk about the liver first. So the glucose would be going into the liver cell through GLUT2. So glucose goes in and then can be phosphorylated. Okay? Now, once that glucose is phosphorylated to glucose 6-phosphate, glucose 6-phosphate does not come back out through GLUT2 because GLUT2 transports glucose, not glucose 6-phosphate. Okay? So why is GLUT2 there? GLUT2 is on liver cell membranes because it has the appropriate characteristics.